The purpose of the Photography of Human Cadaver Prosections project is to produce high-quality digital resources for the anatomy department of Salt Lake Community College, to photographically preserve cadavers whose shelf life is limited, to create representations of the actual cadavers anatomy students study, to illustrate anatomical variability, variability between textbook images and actual cadavers, and variability between one cadaver and another, and to enhance and deepen our own knowledge of human anatomy. The process of photographing cadavers involves 4 separate steps: preparation and creating prosections of the cadavers, the photography itself, post-production, which involves enhancing the images in photo editing software and creating labels that correctly identify the structures anatomy students are required to learn. Not all structures are identified. And finally, the images are uploaded to the Salt Lake Community College Anatomy website for study. Paulina arrives several hours before each photo session, analyzing each cadaver in order to determine which one will be most suitable for study. Salt Lake Community College currently has 8 different cadavers. Effective teamwork between the two of us is a critical factor for achieving our goals. Clear communication allows me to determine the best lighting and composition for the photo session. Next, an actual studio is created for the photo shoot, complete with lighting, reflectors, tripod, and a host of other photographic aids. These are not simple point-and-shoot photo sessions. The first step in the process is preparing the prosections, which requires patience, skill, and advanced anatomical knowledge. Final preparation requires significant attention to detail. The photography begins with the actual shooting and ends in post-production. A vinyl sheet is placed between the cadaver and the surface area to visually separate it from the surroundings. Further subject isolation is achieved during post-production. The third step in the process is post-production. The work is very painstaking and strict attention to detail is mandatory. This is where further isolation and enhancement of the image takes place. Finally, accurate labeling is applied. Only the structures required for student knowledge are labeled. The images are carefully organized in a photo management database. The fourth and final step involves uploading the finished images to the Salt Lake Community College Anatomy website. Here, students can study and review what they have learned in their labs. Each photo shoot, from preparation to final shot, lasts between 4 to 6 hours. 100 or more images are typically created during each photo shoot. Each image processed in post-production requires approximately 1 to 2.5 hours. Not all images are processed. Only the ones we feel best illustrate the structures will be edited. Typically, only 5 to 10% of all images are used. Anatomical variation exists between the textbook and reality. Traditionally, the student must study from a textbook drawing and then locate the same structures on a cadaver. This can be very difficult. Anatomical variation also exists between cadavers. For example, the aortic arch normally has 3 main branches: the brachiocephalic trunk, the left common carotid artery, and the left subclavian artery. The aorta on the right is normal. However, the aorta pictured on the left has 4 branches. The extra branch may be the left vertebral artery. However, this cannot be determined without a deep dissection to see where it attaches to.